Hello everyone, today we will discuss about the composition and structure of the protein. As we all know, protein are made up of different amino acids. So on the basis of this, we can classify proteins on the basis of their orientation and their configuration of the amino acids. So structure of the protein will depend on how these amino acids are present in the chain. Okay, so starting with the primary structure of protein, it's a simple sequence of amino acids without any branching and without any tilting. So now, secondary structure. Secondary structure, it's obtained from the folding of primary structure. For example, if we surround these around our arm, so these will form a helix. That helix is known as alpha helix, and the sheets will also form that are known as beta sheets, and there is a beta turn which is like more advanced from this. So if we further fold these secondary structures, like if we fold these more and more, that will form a fully folded structure, and that fully folded structure will known as tertiary structure. And till this, there was only involvement of a single polypeptide chain. Now if there will be involvement of more than one polypeptide chain, for example, if we talk about the hemoglobin is a 4 polypeptide chain. It's formed of 4 polypeptide chain. Then where more than one polypeptide chain will involve, that will known as quaternary structure. So now talking about the composition of protein. As we all know that protein are made up of amino acids. So these amino acids are linked together by a peptide bond. So we will discuss these peptide bond.
So what is this peptide bond? If we take two amino acids, suppose this is amino acid 1, this is amino acid 2, and we have a carboxylic end of one amino acid and NH2 end of the second amino acid, means the opposite ends of each amino acid. So you can see OH from amino acid 1 and H from amino acid 2 going out, forming H2O. That means if we take the water out by adding two amino acids, as you can see here, the water is carried out, we form here a peptide bond. So peptide bond, we can say, involves the carboxylic carbon of one amino acid and the nitrogen of the second amino acid. So it is formed between carbon and nitrogen. Now this forms a dipeptide, that means having two amino acids. So this bond here formed is the peptide bond. So this is the composition of the proteins. Further, we have already discussed the structures.
Further talking about the nucleic acid, what nucleic acid is. Nucleic acid are a large biomolecules that are crucial in all cells and viruses. They are composed of nucleotides, which are the monomer components. It contains a 5-carbon sugar, a phosphate group, and a nitrogenous base. The two main classes of nucleic acids are deoxyribonucleic acid, known as DNA, and ribonucleic acid, known as RNA. What DNA is? DNA is a double-stranded formation, a double helix. While talking about the RNA is usually single stranded. So the sugar present in DNA is deoxyribose, whereas in RNA it contains ribose. Furthermore, DNA uses the base adenine, thymine, cytosine, and guanine, while RNA use adenine, uracil, cytosine, and guanine. So thank you all of you. This was all for today. See you next time in next video. Thank you.